what I want to talk about today is to follow up only a little bit of the enormous field that was opened up by the development of the electron microscope. And I want to sort of remind you that what happened was not only that the electron microscope was developed and techniques were developed for visualizing materials with it, but a philosophy developed as well, that morphology alone might not be the answer. That in fact, you had to have some understanding of the biochemistry and the processes behind it that eventually will give you a comprehensive view of what cells are like. That at least is the sort of thing that started to emerge during the 1950s. And the people that were very much involved in it were, depending on who you look at, were called mostly by themselves, I think, the Rockefeller group, which is a group of people who ended up in laboratories at the Rockefeller Institute. At that point, it was called the Rockefeller Institute in New York. Interestingly enough, it was set up by David Rockefeller and supported 100% by him. That is, at the beginnings, there was no need for research grants or anything of the sort. The money to do the research was provided by the position that you had at Rockefeller. It's sort of remarkable. At any rate, it became a generative group of people with lots of ideas and lots of process that moved out from there. So in a sense, I'm suggesting you might want to think of it as taking the role of the Royal Society in New York in the 1950s, maybe even in the 1940s. As it turns out, the Carol Moberg, whose book I've indicated over here, Carol Moberg was a graduate student in the late 50s, one of the very early graduate students at Rockefeller. And she has produced a truly extraordinary book, which is called, as you see, Entering an Unseen World. And it's a memoir by herself with extraordinary amounts of detail of the individuals who were involved, as well as personal reminiscences by all of these figures. So there's an essay by George Palade, whom we'll talk about. There's an essay by Phil Siekowitz. There's an essay by Christian de Duve, one after another of the people who were involved in what went on in what became the foundation of the field of cell biology. Well, to a certain extent, this is off to the side of our main thrust of this course, which is just to follow sort of microscopy techniques. But this book, it's available on Amazon. It's available as a Kindle book. It's a truly extraordinary insight, vision into what it was like to be involved in a very active research environment. So with that sort of idea behind us, I now want to mention at least the two major figures that we're going to consider, one of whom we've already met, who was Keith Porter, who worked with George Palade quite a bit. They were very different in personality, but they worked together in a laboratory at Rockefeller, at least initially. Porter was in some ways a mechanical perfectionist. It's an interesting way to think about these things. But remember, he was the one responsible in a lot of ways for making the Porter microtome, the Porter-Bloom microtome, as successful as it was, and developing the techniques for using that microtome. He also developed quite a few techniques for visualizing samples, visualizing tissues. This picture that's on the cover of Carol's book is the first electron micrograph of a cell. taken by Keith Porter. And it was done by growing the cell on what amounted to a formvar surface, exposing it to vapors of osmium, and eventually putting together this picture. It was actually assembled because of the electron microscope that they were using. It was actually assembled out of separate pieces. You can almost see the boundary of one frame here. And another picture that was taken over here, because as it turns out, the electron microscope they were using didn't have a big enough field of view to put the entire thing in at once. Nevertheless, this is the sort of core image to carry around from that. At the same time, George Palade had a strong background in biochemistry as well, and was very interested in the idea of combining biochemical studies with these morphological studies. So the two of them worked together in a couple of ways to try to combine approaches. Palade was also generative in certain, how do I say it? He had a specific interest in looking at an overall dynamic process. So when Porter had seen a structure, what Palade wanted to do was figure out, how does it work? What's going on inside it? How does it fit together? To a certain extent, Porter seemed to be, I won't say content, but he was caught up in what he saw as I think, the structural beauty of the cells of the material that he was looking at. At the time the Nobel Prize was awarded for the development of cell biology, it went to George Palade, as well as Albert Claude, who was a founding teacher, basically, of those two, and to Christian de Duve, who was a biochemist who did extraordinary work in isolating and characterizing lysosomes. Many of you are aware of that interest. Not, in a sense, not part of this thrust of this course. What I want to do is talk for some of the time today about the kinds of anatomy that they found and what they were struggling with to make sense out of it all. So I thought I'd start with an early paper, mostly by Porter, but also Palade, and then Fritjof Sjöstrand, whom you may recall from the last lecture. And what this paper was about, this is in 1953, 1952. The picture on the left shows a hole-mounted cell, something by now you're reasonably familiar with. And you can see in this image, you can see the mitochondria as thin filaments. Remember the old structure of a mitochondrion as a thin filament idea? All of these, and again, conveniently labeled as M with mitochondria. So this is a low magnification image, relatively low magnification image, of a cell more or less dried down. And Palade got very interested in looking at what sections of this tissue would look like, various types of tissue. But he went through a study and came up with these images that you can see here, in which you could see something uniquely sort of mitochondrial, that is, with these cross-membrane structures. Palade was the man who called those structures cristae, interestingly enough. And so he saw these kinds of structures, and frankly, if you ask how would I make a model of what I'm seeing in these things, it's actually quite a challenge. What sort of structures am I really looking at? How many membranes are in this structure? What does my high resolution at that point electron microscope show me and realize that these are pretty limited images. Okay. At the same time this was happening, Sjöstrand was working independently and published his own models, his own images of mitochondria, in which he also saw these same cristae. And the two of them, interestingly enough, Sjöstrand and Palade, came up with rather different models of what they were looking at. So here's a little bit of that history. I'm showing you a much more modern image of a mitochondria now that shows both the outer membrane here and the cristae folding off of the inner membrane. Okay, this is a micrograph taken in 1968, which is, as you can see, sort of 15 years later, from 53 to 68. But Palade looked at the images that he had and created a model in which what he saw, he said, was essentially that kind of image. That is, that there was an outer membrane, and then that there was an inner membrane, and the inner membrane was thrown into these folds. You've seen this diagram forever. On the other hand, Sjöstrand looked at very similar images and just simply said, no, that ain't so. And so what he drew, instead of that model, you can see in this diagram, was basically an outer membrane, an inner membrane, and the cristae as separate internal sacs. And so this kind of difference of opinion actually was impossible to resolve based on the equipment that they had available. But as it turns out, these people who were involved ended up with a certain kind of personal conflict. As I mentioned in the last lecture, Sjöstrand and either Palade or Porter didn't get along that well. And so in this wonderful book by Carol Moberg, there's a report, which I've copied over down here, which basically says that when Porter and Sjöstrand were at a meeting, Palade wasn't able to be at the meeting for whatever reason. But Porter wrote to Palade that, you see, our friend Sjöstrand is made up of worse material than I suspected, worse even than the one implied in your comment about paranoia. I'll take care of his desires in the next paper, but probably he won't like that either. I mean, obviously, Porter, who has a kind of an acerbic style anyway, really didn't have a very high opinion of Sjöstrand's personality. As it turns out, he respected his work quite well, and he commented in a number of things later on that not only Sjöstrand, but his student, who was a man named Johannes Rhodin, or Rhodin, R-H-O-D-I-N, really was coming up with very, very impressive images. But what differed was the way that they interpreted them. So it's kind of an interesting thing to realize that these people were, in a sense, blind men struggling to figure out what an elephant was and how the elephant was structured. You can look at one corner, you could look at the other, but since there was actually no good model of what was going on in the mitochondria, that made it particularly more difficult. It took until the late 60s for Peter Mitchell's models of the osmolarity effects, right, of the chemiosmotic nature of mitochondrial production, of to make you significantly aware of the role of the membrane systems in the economy of the mitochondria. Nowadays, we just sort of talk it all out as established fact, but it took quite a while for these people to actually come to an understanding of what was going on. And by then, certainly Porter and Palade had passed on from the mitochondrial interest, although Sjöstrand retained his interest quite long and was pretty convinced that the cristae were not being treated properly in the other models, even later on in his life. And one of the interesting things, of course, here is how long these various people have lived. So they've had a long time to think about this work. So let me move on to the other structure that's even more puzzling and more bizarre. This is the one, I'm using the same illustration as a starting point from Keith Porter's initial paper on the structure of cells, intact cells. He then went ahead and added a second paper in which he described what was then called basophilic structures in cytoplasm. And it's an interesting sort of thing. People had discovered, as a background to this, people had discovered that even with light micrographs and light microscope images, that certain regions of the cell would stain with a dye that's specifically bound to acidic material. So it's basophilic material bind to these structures. And it was used actually as a way of thinking about nucleic acid, because nucleic acids were obviously acidic. And it was this dye, these dyes that would bind to the acidic materials. So it's certainly bound to the nucleus, but interestingly enough, bound to the cytoplasm. You couldn't tell much about the way it bound to the cytoplasm, but there seemed to be these sort of basophilic regions within the cytoplasm cells. And people also had this interesting term of ergastoplasm, where people were trying to struggle with what is the real structure of a cell. Is a cell a fluid vesicle? I mean, a large fluid container with some of these things floating around in it, or in fact, does it have some internal structure? And one part of that internal structure was this material that was basophilic and was called in this peculiar term ergastoplasm. Well, if we take a look at an enlargement of an area of a cell like this, or if we look at the edge of a cell like this, we see the mitochondria first, and then we see this network. See it here. Okay, and this was seen in the paper I'm showing you. This was all described by Keith Porter. But he saw this thing as a network, and he was able to enlarge it. This is a shadowed version. The one that's called Figure 7 here was a shadow cast version of also a dried down cell. And once again, he saw this material between all the mitochondria. And well, in all honesty, this is the sort of thing that gave electron microscopists a bad reputation, or a reputation for having an extraordinarily good imagination. How could something like that end up having a real structure and being a real structure? Okay. Well, one of the things that Porter tried to do was he was aware of a study that showed that you could isolate material. You could grind up cells and isolate a population of membrane material that also seemed to have this basophilic property. Okay? And he got a sample of it and put it in the electron microscope and saw this. You can see what you want out of it. I think his idea was, look, we're seeing little strands of material. There may be globular material, but there's something in there that maybe is similar to what he's seeing in the cells. As I said, this is all the sort of thing that requires an enormous sense of imagination in a certain way. And so even Porter was aware of it. So there's this wonderful line that Porter apparently introduced cell structures in lectures by saying, and you may not be able to read this, that some of my acquaintances think less highly of the ER than I do. They facetiously refer to the stuff as portoplasm. So there we are. It turned out actually to be paladeplasm. And we'll talk only a little tiny bit about it because Porter stopped looking at this material and George Palade picked it up and became very interested in what this membrane material really was and what it could function as. And what Palade saw and concentrated on, I don't have a good image of it to show you at the moment, but what Palade saw was that these membranes also seemed to have particles on them. At least many of them did. And so now people started to divide up this ER material into the ER that had particles, the ER that didn't, and what became known as smooth ER and rough ER. And then what Palade showed was that the ER was in fact studded with a structure that was now what we know as ribosomes. So all of this work started from these kinds of electron micrographs. What Palade eventually did was he worked with a biochemist whose name is Phil Siekowitz, who worked to show that those microsomes, those membrane fragments, arose from the ER and did contain what then became known as the ribosome. So it's sort of remarkable stuff. As I said, the history of cell biology, of which this is kind of just a little bit of an insight, okay, of the way these people were digging around in an area that was brand new. I mean, they had the background of knowing something about light microscope studies, but by and large, what they were seeing had never been seen before. So the parallel, to go back to the Royal Society, right? The parallel with Robert Hooke and von Leeuwenhoek is that now, as resolution has gotten even better, and preparative techniques have gotten even more elaborate, one starts to be able to look again at a new world. To me, that's kind of an amazing historical view. And what I want to do now is show you that in another 10 years or so, the techniques had developed to the point where it was possible to produce basically an atlas of images that served as a basis for quite a bit of subsequent histology. And just to give you an example of a couple of these images, here's a couple of examples. The book was put together by Keith Porter and initially a postdoc in his lab named Mary Bonneville, and they worked together to assemble their very best pictures. And apparently, Porter was something of a maniac in making sure that these things were really crystal clear and beautiful images. And as you see, the products are are quite impressive. So this is a bit of, I think it's a liver cell, and you can see within it all these interesting structures. So now you can see mitochondria in different configurations. So all of these things, right? And that you certainly don't see them in that sort of awful way that they appeared, say here, one more, right? When the sections were too thick and the stain wasn't well developed. Right now, the details are pretty much more impressive. So you can see a different tone. The other thing that Porter and Bonneville introduced, which I find very interesting, was this sort of grayish tone to the images. And what that did was it sort of filled in the model that cells were full of material. So it's a kind of an interesting way of thinking about it. Well, here's a little bit of endoplasmic reticulum right over here as sheets of membrane. And we now know that they're both sheets of membrane and tubular structures, but you can see them scattered through the cytoplasm here. What I'm seeing as this dark material is glycogen granules, which is why I say that this is most probably a liver cell. And up here, you can see the membrane of the cell and a junction to the next cell over. So it's kind of extraordinary how all of this is present in almost every cell that you look at. There's a common structure. These are microvilli here, extending out. So let's take a look at another image. This is presumably from things like the trachea, from those epithelial cells of the trachea, because what you see in the cells here are these ciliary structures. These are the cilia sticking out into the space. This is where they would, in your trachea or in your lungs, where they would be pumping the mucus around and away. Okay, you still have mitochondria, you still have endoplasmic reticulum, and some of the ER as indicated here is smooth ER, while others seem to be more like the rough ER that we're familiar with, different cell types among others. Okay, and here's this wonderful example of what a cross section through these cilia look like. And so in the one picture, you get a sense of the extent of cilia and also what it's like to cut through them, cut across them. And here you see very clearly that 9 outer doublets and the 2 central doublets. Whereas here, it's a little in these longitudinal sections, like this one over here, it's a lot harder to see that sort of structure. Okay? But pretty impressive to realize that an atlas like this was generated maybe 15 years after serious electron microscopy began. So here's another image. This is an intestinal cell. What I have here, what's visible, is this enormous array, again, of microvilli. A mucus producing cell over here. Mess it up. And this very elaborate membrane, starting with junctions here, and then with all of these interfolds like this between the two adjacent cells. Actually pretty impressive. And cheek cells themselves show a structure like that. And the idea is that the two membranes, here's one membrane of the cell, here's the other membrane of the cell. In a situation like this, where it's really quite essential for the survival of the organism that nothing leak from the intestinal acinus between the cells, digest the cells themselves, so that what happens is the membrane not only exists, but it sort of folds itself around a little bit. So the membrane of the adjacent cell is locked into place. Okay? Which is pretty, again, a remarkable bit of structure. So there's, again, a whole world in here, and we're not going to go through all the details, except I can point out that in these particular sections, you now see the euchromatin and the heterochromatin, as well as nucleolar structures very clearly, both of these. Porter and Bonneville put this thing together. There are about 50 separate images in this book. And each one of them became a revelation. So I'm not going to go through the entire thing now. But at some point, it's one of the books that's worth looking at just to get the aesthetics of how exciting this cellular structure can be. And that's independent of the mechanisms, of the processes. So this is the kind of thing that Porter is sort of best known for. While what Palade did, and what probably justified the Nobel Prize to him rather than to Porter, was that he was very interested in going beyond the morphology and into the mechanism. And so that made quite a difference, certainly for the people doing the Nobel Prize evaluation. Okay? And the last picture I want to show you is kind of an invitation to think about problems that we're going to actually solve in the next lecture. And this is a classic image, if you will, of two plasma membranes. You can see them sort of at low magnification here. And over here in much higher magnification. Okay? And the question is, what is one seeing when one looks at this membrane? But if I take a single one of these images, or try to make a diagram of it, what was originally done was people said, well, a membrane consisted basically of two sides, like this. That's what you see in this image. The two dense lines with a lighter space in between. Except, one could argue there are all these funny little interferences in this membrane. And the question is, do those represent real structure? For instance, if I look at this little segment here, could I argue that what I'm really seeing is something that looks sort of like this? Lots of globular material put aside. Okay. What I want to do is lead you through to next lecture, which will have to do with the development of the technique of freeze fracture and how a whole other way of looking at the structure of membranes emerged and revolutionized again how we think about those structures. Just one more side issue, just to remind you that what you're seeing in this corner of this image is a bit of endoplasmic reticulum with ribosomes on it, right? And what's peculiar is to realize where we are in this image. Okay? point out, if this is the membrane of the ER, and there's another membrane here, the dots that you see are actually the ribosomes in the cytoplasm. So all of this material here is the cytoplasm of the cell. And these little spaces are the containing areas, the cisternae of the ER. Just something to think about again. So for next time, we'll put some more time into this idea of what is in fact the structure of the membrane as we see it through technology, through the procedures and techniques that let you examine it. Okay, we'll stop here.